Hello everyone, thank you for joining us today. My name is Sarah and I normally see you at the Museum of Zoology, where I work in the public engagement team. I'm very pleased to welcome you to week 4 of our 4-week course, where we have been exploring the animal kingdom and real conservation stories together each week. I'm joined today by Giacomo Gattoni. Giacomo is a PhD student in the Department of Zoology, studying evolutionary biology. He's in the museum gallery now, where he's going to give us our talk. He's studied echinoderms in the past and will be focusing on this group of animals today. So Giacomo, what is an echinoderm?
Hello. So echinoderms are a group of marine animals, so animals that live in the sea and the ocean, and it actually live in most seas and at most depths. We can find them basically everywhere. And some of these are actually quite familiar, animals that we're all quite familiar with, like starfish and sea urchins, and then there are others like sea cucumbers and feather stars, so a little bit strangers. So they're all marine animals, and we have actually quite a good collection here in the museum of, as I said, all the different kinds. So we have some sea urchins right here, some sea cucumbers, with, again, they're, as the name suggests, they're sort of cucumber shape, and starfish. And all of these animals basically are part of this group because they have the same body plan. So the same way basically of building a body. And to explain the body plan, so the structure basically of the sea urchin, we're going to now go to my laboratory and have a look at the live starfish that we got there to help us understand the quite strange body that these animals that we will see have.
So here in the video, we see a live common starfish, which is actually a starfish that lives in the seas around the UK. And again, everyone is quite familiar with the shape, but the key point here, if we think of humans or most other animals, like again, a cat or a dog, we can think of it as having a left and a right side. They are quite recognizable. So if we basically draw a line in the middle, we will have two sides, one left and one right, that are specular, symmetrical. And this is called bilateral symmetry. Instead, starfish and the other echinoderms are unique in having a radial symmetry with 5 different units that we can see here repeating around the body. So basically, here, if we draw a line on one side, we will be dividing the animal in 2 halves. But if we draw another line from this side, it will also draw the animal in 2 halves. And we can do this 5 times and we will always get the symmetrical body.
So we are now back in the museum gallery to take a look at another type of echinoderm. What's the next animal?
So we've seen the starfish, which we can say is the most famous echinoderm, but we have quite another popular one, which is the sea urchin. And as I said, we have quite a few sea urchins here. And what we can see in the sea urchin, even if externally, when we look at it, it looks quite different from a starfish, all like sort of spherical with all these spines pointing out of it. We can actually see, though, by looking at it, that we can see the same structure of the body that we see in the starfish.
So yes, I am a bit more familiar with the sea urchin than I'd like to be, having accidentally walked across one whilst walking around in the shallows on a tropical beach. So can't recommend that, but they most definitely have those long spines and yes, like to move around on the floor of the sea.
But yeah, exactly. You don't want to step on one of them. And yet, even if the structure again looks so different from the starfish, if we look inside, and in particular at the skeleton, we can see again this sort of the same type of symmetry, sort of radial symmetry that we see in the starfish. And in particular, we have a picture of the skeleton in your booklet in the page with the green band on the side. And we can see there again in the structure of the skeleton, we can see always these 5 rays. And in all the species of sea urchin that we look, we always see these 5 rays, which again show us that it's the same pattern and the same symmetry that we see in the starfish. So we've seen that all these animals share the same body plan, the same quite strange and quite unique body plan, as I was saying, and we were seeing. And this sort of was very curious for evolutionary biologists. We're trying to understand where did this shape come from? How did this very unique body plan evolve? And to do so, to try to understand this, scientists started to look at the development of these animals. So the process by which you get from basically an egg to an adult organism. And if we look at the development of starfish and sea urchin and the other echinoderms, we see that they have what is called a larval stage. So instead of going directly from an embryo to an adult, they have an intermediate stage that is quite different and that can already feed and move, very similarly to the frog that has a tadpole stage, which again is quite different from the actual frog, but can still already move and eat. And then basically from that, the adult form will develop. And sea urchins are very similar. So each one of the different types of echinoderms have their particular larval stage. And here we have a picture of a model of the larva of a sea urchin that we have always in the page with the green band, which is called a Pluteus.
Oh, Pluteus, that's almost sounds sweet, doesn't it?
Yeah.
So if we look at the model of the larvae in our booklets, like you say, with on the page with the green bar on the side, it looks really different to the adult. And in fact, we're talking about this radial symmetry where we can't draw a line down the middle. Actually, I think I could draw a line down the middle of the larva.
Yes.
So how did it evolve to be that the larva looks so very different to the adult?
Yes, so that is exactly the key point that scientists discovered by studying the development of echinoderms and by being so sort of puzzled by how strange they looked. That if we go and look back at the larva doesn't have this sort of radial symmetry, but have a symmetry just like ours. So in which we can draw a line in the middle and define a left and a right side of the body. And this basically showed us that the ancestors of echinoderms and that the ancestors that we share with echinoderms all were like us. So had a left and a right side of the body, just like us. And then during only the evolution of echinoderms, very gradually, the adult shape changed more and more until it became this sort of weird, symmetry of 5, in which we have these 5 different rays that are all a repetition of one another, basically. And so we can see how by studying development of these animals, we can actually reconstruct their evolution in the past.
Oh, wow. So we can learn so much from the larvae of echinoderms about evolution and about evolutionary biology. What else can we learn?
So actually, echinoderms are used as a good model because of some of for other types of studies, because of the characteristics, especially of their larvae. So the adult starfish and sea urchin kind of moves on the sea floor or on rocks, right? So walks basically around the sea floor. Whereas on the other hand, the larvae are very, very small and swim around in the water column. So they have little cilia basically that beat and propel the larvae in the sea. And this means that while they're doing this, they're also eating. So they have a sort of little mouth in the anterior part of the body. And the way they eat is just they basically filter the water. So they eat all the food particles that are present in the water. And this is my way of feeding is called filter feeding, because again, they're sort of getting all the food particles in the water that pass through them. However, this method of feeding also means that the larva would come in contact with any kind of substance that is present in the water. And so this makes them models that are very good models that are used in ecology to understand in particular the effect that some chemicals and some other types of pollutants can have on marine life. And for example, in the lab in Milan where I was studying the evolution of echinoderms, we were also studying the effect of a particular class of contaminants that are present in the water, which are microplastics. And microplastics are these very, very, very tiny pieces of plastic that are released in the ocean, sometimes when bigger pieces of plastic break, or in some cases that microplastics are produced and then just released in the water as part of other types of products. And the point is that these are, again, as I said, very, very tiny, so that even very small animals like the sea urchin larvae can actually ingest them. And in the laboratory, they looked in this study at the effect of these microplastics. So they raised this larvae in the water full of microplastics, and they saw that first of all they were able to inject it. And we can actually have a picture of this in the right side, right page of the booklet, the one with the blue bar. Here we can see two types of Pluteus larvae, so the sea urchin larva. One raised in clean water and one raised in water with microplastics. And we can see that the stomach of the second one is full of the red microplastics. So this means that the animals eat them as they're just filtering the water. And this fills their stomach and probably then doesn't allow them to have other food in it. And in fact, the second finding of the study was that after ingesting microplastics, the larvae were smaller than the ones that were raised in clean water. And again, this is probably because of this effect, of the fact that the Pluteus, the larva didn't have space in the stomach to ingest, to digest other food, basically.
Mm-hmm.
And
that's really interesting. So the fact that the larvae of echinoderms don't really have a choice in what they consume, because as you say, they are filter feeders. They will eat whatever is floating around in the water with them. They can actually help us to see when waters have pollutants in them, and pollutants that we might not be able to see with our eyes, especially, and see the effects of them on the wider environment as well as echinoderms as animals too. So,
yes, that's exactly right. So these larvae and other types of filter feeders, so animals that filter the food that they're eating, are often used as indicators of the quality of the water that they are in, basically. And in the case of the sea urchin larvae, this is particularly good because they are produced in great amount. So when the sea urchin releases their eggs, they release thousands of them at the same time. And so we have a lot of animals that can be used to check and understand the quality of the water. And then they're also very, very, very small. So even small differences or small concentrations of these pollutants can have an effect on these animals. And so we can actually, check with a very fine, in a very fine detail, the state of the water, and also, of course, what we can then do to prevent this from damaging the marine animals.
Excellent. So they are a really useful species for pets as well. Well, thank you very much for your time today, Giacomo. We are now going to go to our live segment where our audience can ask any questions that they have about echinoderm, starfish, sea urchins. So for now, thank you very much.
Thank you. I look forward to it.